Okay, let's start the class. Meanwhile, others can join. Uh, so, hi everyone. Today is the 12th and the last problem solving session for the course Environmental Chemistry and Microbiology. The course instructors, as you know, are Professor Anjali Pal and Professor Sudha Goyal from IIT Kharagpur, and I am Shrija Ghosh, a PMRF from IIT Kanpur. So today's session is all about different biogeochemical cycles, and then we'll study about more about metabolic diversity. In the previous session, also we covered a few points regarding metabolic diversity. So we'll solve the questions related to all these topics, and I'll request all of you to answer the questions in the chat box, and we can discuss if your answers are right or wrong. Uh, I would request all of you, if you have any doubt at any point in any of the questions that I am discussing or in any of other question, please let me know. I'll try to help you out as much as possible. Okay. One thing before I start, in the previous session, somebody asked me about a question in week 10's assignment, the limiting nutrient one. So that one, it the answer should be both A and B, macro and micronutrients. So I think the answer key is wrong. So I would request you to talk to the NPTEL team regarding that. Okay.
No, we have already mailed. So when we will get our answers correctly.
You had mailed. You did not get a
Yes, ma'am.
response.
Yes, ma'am.
Okay. I because we are not associated with the NPTEL team. We are just like we are students of IIT, and we are just taking your doubt clearing sessions. I'll see if I
Ma'am,
can contact
ma'am, your
somebody.
voice is
Yeah.
cracking.
Yeah, just a second. Can you unmute your mic, Rudrat? Sorry, mute your mic. Thanks. Uh, I was saying, is it fine now? Is my voice clear or better?
Yes, yes, ma'am. This is
Huh.
clear now.
Yeah. So I was saying, actually, we don't have any direct contact with these NPTEL portals and the people handling the answer keys. So I'll see if I can contact somebody, because we are students of IIT, and we are just asked to uh, we have been asked to take your problem solving sessions and doubt clearing sessions basically. So I'll try to contact somebody, and you also wait for the replies. Let's see. Let's hope for the best, because generally they do clear like correct the answer keys if the keys are delivered wrong. So I'll try to see. We have one more session next Tuesday. That would be our last session. So I'll take a revision of all the topics that are there for all 12 weeks topics. So there I would, like, by that time I would try to get this rectified. Okay.
Okay, ma'am. Thank you, ma'am.
Yeah. Okay. Let's start today's class then. Starting with the first question. Consider the following with respect to carbon cycle. Consider the following with respect to carbon cycle: ocean, forest, soil, and abandoned mines. Which of the above mentioned cannot act as carbon sinks? Which of the above mentioned cannot act as carbon sink? So you have four different options: ocean, forest, soils, and abandoned mines. Now, out of these four, which cannot act as carbon sinks? So, first option is 1 and 2 only, that is ocean and forests only. Second option is 1, 2, 3, and 4, all four of them. Third option is uh, third and fourth only, which is soils and abandoned mine. And option D is fourth only, that is abandoned mines. So all of you write your answers in the chat box. I would request all of you who know the answer should write the answer, so that I'll get an idea of how many people are like who are understanding this, and if somebody has doubt, please let me know. I have only 3 answers in the chat box. Anyone else? Anyone else wants to answer? Okay, all 3 of you are saying option D, fourth only. Let's see the correct answer. Fourth only means abandoned mines. That is the correct answer. So, what are carbon sinks? Carbon sinks basically are the places that can absorb more carbon from the atmosphere than it would release. For example, plants absorb carbon dioxide from the atmosphere, so it's a form of carbon. Oceans are absorbing carbon, soils are absorbing carbon. So, the amount of carbon that is absorbed by uh, these plants, oceans, and soils, uh, it is more than they would release the carbon. In contrast, the carbon sources. So there are two terms: carbon sink and carbon source. Carbon source are the ones which can release more carbon than it can absorb, like volcanic eruptions. Volcanic eruptions release carbon to the atmosphere more than it can absorb carbon. It releases carbon. Then burning of fossil fuels, you all know burning of fossil fuels are ultimately releasing carbon in the atmosphere. They are increasing the carbon load in the atmosphere, right? So similarly, abandoned mines can also act as a carbon source. They can release carbon in the atmosphere. They cannot act as a carbon sink. Okay, abandoned mines can be turned into a carbon sink when you try to grow, like if you make it fertile somehow, add soil and then add plants. If you can turn the abandoned mine into a carbon sink, that is basically a measure towards protecting the environment. Otherwise, these all the carbon sources are really harmful for the environment because they are adding up to the carbon uh, of in the atmosphere, right? So carbon load needs to be reduced in order to reduce your environmental problems like global warming. Okay, so we need more and more carbon sinks in the atmosphere and less carbon sources. Is this clear? If anybody has any doubt or any questions regarding this, please ask. Okay, I'll move to the next question. Question number 2. What organism represents the symbiotic relationship between the roots of a plant and fungi? What organism represents the symbiotic relationship between the roots of a plant and fungi? Option A, legume. Option B, mycorrhiza. Option C, black root. Option D, lichen. Which organism represents the symbiotic relationship between the roots of a plant and a fungi? So, what would be the answer? Option A, legume. Option B, mycorrhiza. Option C, black root. Option D, lichen. Write your answers in the chat box, all of you. All of you write your answers. Again, I have only 3 answers in the chat box. 4 answers. Symbiotic relationship between plant and fungi. All of you are saying option D, lichen. Are you sure about it? Mubani wrote option B and then changed her answer to option D. Anyone else wants to answer? It's plant and fungi. Anyone else wants to take a try? Take a guess? Okay, the correct answer is option B, mycorrhiza. So, mycorrhiza is the symbiotic relationship between roots of plants and fungus. So, the basically symbiotic relationship means both the parties are being benefited uh, when they are living together. Okay, so this plant, the roots of the plant and the fungus, they are living in close proximity, and both of them are taking advantage of each other. Okay, they are, both of them are getting benefits from each other. How they are getting benefits, let's see. So the roots of the plant are giving a place for the fungus to live. The roots of the plant and the plant itself, they are providing simple carbohydrates, inorganic nutrients. So all these things are provided by the plant, by the roots, by the soil around the roots, and the fungus is easily using these carbon sources and nutrients to grow. So basically the fungus is getting its food from the plant. In return, what the plant is getting, since so fungus generally has a mycelial growth. Okay, so mycorrhiza is a type of fungus which has a mycelial growth. What do we mean by mycelia? Mycelia are the fine thread like structures. So if you have commonly, you must have seen fungus, white colored fungus growth on bread, or like whitish greenish cotton like fungus on some food items. uh, I'm hoping all of you have seen such fungal growth on different food sources. So, in those cases, you have seen the fungus looks like a cottony mass, right? So, that cotton, if you see very closely, it has fine thread like structures, and all those threads are called mycelia. So, there are many fungus like that which have a mycelial form of growth. Not all fungus have mycelia. Some fungal cells are unicellular, like yeast, and some fungus make proper fruiting bodies, like mushroom. But there are mostly there are fungus which make mycelia, these thread-like structures. Now, what these thread-like structures are doing, they are growing over the roots and increasing the root surface area so that root can absorb more and more nutrients. What is the role of the roots in a plant? The role of root is to absorb nutrient from the soil and deliver it to the rest of the body of the plant for doing all the things that it has to do, for doing photosynthesis, for all the biochemical processes that are going on inside the plant's body. Roots are the ones that are absorbing the nutrients for it from the soil. This fungus is growing on the roots, and since it has a thread like structure, the thread like structure is basically increasing the surface area of the root itself. Root itself is a thread like structure. The reason that roots have that hairy thread like structure is to absorb more and more nutrient. And imagine over that, if more uh, thread like structures start growing, then it is basically increasing the absorptive area of the plant root system, right? So basically the fungus is getting food from the plants, from simple carbohydrates and nutrients, and the plant is getting help in absorbing more nutrients from the soil uh, with the help of fungus. Okay, so this symbiotic relationship where both of them are getting some benefits, that kind of relationship, it's called a symbiotic relationship, and between a plant and a fungal species called mycorrhiza, it's a very unique relation. Okay, I hope that's clear. Anybody has any doubt? If you have any question or any doubt, please let me know. I'll move to the next question meanwhile. Question number 3. Oxidation of ammonia into nitrites is carried out by. Oxidation of ammonia into nitrites is carried out by option A nitrobacter, option B enterobacter, option C nitrosomonas, and option D rhizobium. Oxidation of ammonia into nitrites. We have discussed about this in one of the previous session also, where we were discussing about nitrogen cycle. So in this session, since this session is about biogeochemical cycle, we will again discuss that in detail. So, but the name of the bacteria we had discussed in the previous session. So all of you should be able to answer this. Which of the following helps in the oxidation of ammonia into nitrates? Nitrobacter, enterobacter, nitrosomonas, rhizobium. I have only one answer in the chat box. What about others? Don't remember? We have already discussed this. This everybody should be able to answer. I have 3 answers in the chat box. Anyone else wants to answer? No one? Okay, let's see. The 3 of you are saying option C, nitrosomonas. That is the correct answer. So, nitrosomonas is the bacteria that converts ammonia into nitrite. So, do you remember what is this process called? Conversion of ammonia into nitrites and then nitrates. Do you know the name of the process? Anybody? One of you can write it in the chat box. What is the process where ammonia is oxidized into nitrites and then nitrites are oxidized into nitrates? The process is called. It's a one of the reactions that happen in nitrogen cycle, right?
Ma'am, is it nitrification?
Yes, it is nitrification. So the process of oxidation of ammonia into nitrites and then nitrates, that is nitrification, and nitrosomonas carries out the first step, that is ammonia to nitrite. All these bacteria that are there, they have some or the other ecological significance. So nitrobacter is the one that can convert nitrite to nitrate. That is the second reaction of the nitrification. So nitrification has two reactions. It starts from ammonia. Ammonia gets oxidized into nitrite, and then second step is the oxidation of nitrite into nitrate. In the previous class, I had explained how many electrons are involved in this whole nitrification process. So there also I had shown the reactions. If you haven't seen, you can revisit the video or the slides to see. Okay, so nitrobacter is the bacteria that uh, plays the role in the oxidation of nitrite into nitrate. Then enterobacter, the second option that was there, it's a coliform bacteria, and it is mostly pathogenic, and it causes UTI and respiratory tract infections. Okay. Then nitrosomonas, we already discussed. It uses ammonia as a source of energy in the presence of oxygen, and it utilizes carbon dioxide as a carbon source. So something that is utilizing carbon dioxide as a carbon source, what is it called? This also I've discussed in the previous session, right? If some organism is using carbon dioxide as a carbon source, what is it called? You've forgotten everything. If something is using carbon dioxide directly as a carbon source, it is called an autotroph. Okay. If it is using organic carbon as a carbon source, it is called heterotroph. We had discussed all this, right? And there are other bacteria that can catalyze this reaction, the oxidation of ammonia into nitrite. They are all these bacteria are called ammonia oxidizing bacteria. Okay, so apart from nitrosomonas, nitrosococcus and nitrosospira, they can also catalyze this reaction. Then the fourth option was rhizobium. Rhizobium is actually a gram negative soil bacteria that can fix nitrogen. So if you remember from the previous session also, when we discussed this nitrogen cycle, I told you the ammonia is converted into nitrites and then nitrates. That was the nitrification process. Nitrates are converted back to nitrogen. That was the denitrification process. And the nitrogen is a gas, it goes back to the atmosphere air, but there are certain bacteria that can fix that atmospheric nitrogen into ammonia. So nitrogen is again converted back to ammonia. So rhizobium is one such bacteria that can fix nitrogen, that can fix atmospheric nitrogen and convert it into ammonia. And these bacteria are known to form symbiotic relationship with the roots of the legumes, enabling nitrogen fixation. So like we read about mycorrhiza, where fungus and the roots of the plant had a symbiotic relationship. Similarly, these bacteria, rhizobium, they have a symbiotic relationship with the roots of the leguminous plants. So legumes are the ones which we call pulses. The dried versions are called pulses. So all the pea family plants basically, they come under this category of legumes, and their roots have these structures, spherical structures called nodules, in which these rhizobium bacteria reside, and they fix the nitrogen. So they are also taking benefit from the plant by using the carbohydrate sources from the plant, and they are fixing the nitrogen, and the plant is using the product of the nitrogen fixation. Okay, so they both of them are getting benefited from each other. So it's a symbiotic relationship. So these are the processes of nitrogen cycle. So nitrification, as I've already talked about, so ammonia to nitrite and then nitrite to nitrate. Okay, so ammonia to nitrite, there are different bacteria that can do that: nitrosomonas, nitrosopumilus, then in the previous slide we saw nitrosococcus. So all those bacteria can convert all these bacteria are called ammonia oxidizing bacteria. Okay, and then we have nitrite to nitrate, which is done by nitrobacter mainly. Then the denitrification process, where this nitrate is converted back to nitrogen that goes into the atmosphere. It is carried out by bacillus, pseudomonas and paracoccus. Then this nitrogen is fixed by certain bacteria, and ammonia is the product. So there are different nitrogen fixation processes. So if the nitrogen fixation is happening in presence of oxygen, that is aerobic nitrogen fixation, that is carried out by cyanobacteria and azotobacter. If it is happening in the absence of oxygen, it is called anaerobic nitrogen fixation, and the bacteria are Clostridium and Methanobacterium. Then symbiotic nitrogen fixation, like I already explained about rhizobium, there are other bacteria called bradyrhizobium and Frankia. They also do the similar kind of symbiotic nitrogen fixation. Then there is one more process called ammonification. So all these nitrogen fixation the aerobic, anaerobic, and symbiotic type, they are actually fixing the nitrogen that is present in the atmosphere, that is present in the air. Okay, that they are using up the nitrogen in the air and converting it into ammonia that can be used up by different other bacteria and plants. Okay, another process called ammonification, that is also nitrogen fixation, but there the source of nitrogen is not air or atmosphere. The source of nitrogen is from the organic waste matter. So if there is some organic waste matter in which nitrogen is present, that nitrogen is directly fixed by certain bacteria like bacillus, pseudomonas, clostridium, streptomyces, and they can directly convert that nitrogen into ammonia. And then there is another class of bacteria that can directly convert the ammonia and the nitrite into nitrogen. So the denitrification process was similar to denitrification process where nitrates were converted into nitrogen. Here the before nitrates, what do we have? Nitrite and ammonia. So nitrite and ammonia can be converted into nitrogen, and that process is called anemox. Okay, and the bacteria that does it is Brocadia. I hope all these processes are clear now. So this is actually very important. So you all should be able to at least remember the process, remember the reaction, and at least one bacteria, one representative bacteria. So you know the first step of nitrification, that is ammonia to nitrite. All the bacteria, mostly all the bacteria that are starting with nitroso prefix, that are basically involved, that those are basically the ammonia oxidizing bacteria. Then we have the nitrite to nitrate, that is mostly done by nitrobacter. So if you remember one example, that is more than enough. Okay, so some important rhizobium, you should remember about rhizobium. It, because the questions about rhizobium, it's very common, the symbiotic relationship between plant and bacteria. So those things, some of the examples are actually very important, so you should remember what is anemox. Okay, anybody has any doubt or any questions? If you have any question or any doubt, please write it in the chat box. Okay.
Ma'am, can you explain the processes just in short once again?
Yeah, so the process actually starts from nitrogen fixation, if we see, because nitrogen is there in the atmosphere. So there are different classes of bacteria that are using that atmospheric nitrogen and converting it into ammonia. So if it is doing it in presence of oxygen, it would be aerobic. If it is happening in absence of oxygen, it is anaerobic. And then there are certain bacteria that are living in close proximity with other organisms, other species, or other uh, class of organisms, and they both are helping each other to survive. So that is a symbiotic relationship. So there are certain bacteria that are symbiotically fixing the nitrogen. Okay, then there is another process called ammonification, where the nitrogen that is present in the organic waste matter are fixed into ammonia. So once all the nitrogen are converted into ammonia, this ammonia can be now used by certain uh, other bacteria. So ammonia, basically it can take up protons, water and protons are already there in the environment. So ammonia or ammonium ion will be oxidized into nitrites by ammonia oxidizing bacteria. Okay, and nitrites can then further be oxidized into nitrates by nitrobacter. These two processes combined are called nitrification. Okay, so after nitrogen fixation we have nitrification. Nitrogen fixation, we had nitrogen to ammonia. Nitrification, we have ammonia's oxidation. So by oxidation, if you see the oxidation state of the nitrogen here, it would be -3. And then if you see the nitrogen's oxidation state in nitrite, it would be -4, and then this would be +3. Okay, and this would be +5. So basically the oxidation state of nitrogen is increasing. That means the nitrogen in the compound is getting oxidized. So oxidation process is nitrification. Once completely oxidized form of nitrogen is there, it is again reduced to uh, atmospheric nitrogen, whose oxidation state is 0. So this is a reduction process, and it is also called denitrification, because now the opposite process of nitrification is happening basically. So it is denitrification. Again, it would be a stepwise process, but it's generally shown in a direct reaction, nitrate to nitrogen. So again we came back to nitrogen, and that nitrogen can again uh, be fixed by different bacteria to into ammonia. Ammonia can be oxidized into nitrates, and then nitrates can again go back to nitrogen. So this is the cycle keeps going on. Other than that, there are other side reactions that happen that are called anemox. Okay, is this clear now?
Yes, ma'am. Thank you.
Okay.
Ma'am, could you please show us the previous slide?
This one or the question?
Yes, ma'am. Thank you, ma'am.
Okay. Okay. I can move to the next question now. Question number 4. If anyone still has any doubt, please write it in the chat box. Okay. Question number 4. What organism is the result of a symbiotic relationship between a fungus and an alga? So now we have seen fungus and plant, bacteria and plant. Now this is about fungus and an alga or algae. Option A, lichen. Option B, mycorrhiza. Option C, legume. And option D, rhizobium. Now this should be answered by everybody, because we have already discussed what mycorrhiza is, what rhizobium is. What is the symbiotic relationship between fungus and algae? Why only 3 people are answering continuously? All of you write your answers. By this time, you should know what should be the answer to this question. Okay, I have 4 answers. 4 of you are saying option A, lichen. Anyone else wants to answer? Okay, so the correct answer is option A, lichen. So mycorrhiza, we have already discussed, it's a symbiotic relationship between plant and the fungus. Legume is just a class of plants. I've told you the plants which are from the pea family. They are leguminous plants. And then rhizobium, we have discussed, it's a symbiotic relationship between a bacteria and a plant. Okay, lichens are the ones which has a symbiotic relationship, which is a symbiotic relationship between a fungus and an algae. So how both of them are helping each other? Basically, algae are generally photosynthetic organisms. Okay, so they can use the carbon dioxide, they can use the sunlight, and easily make uh, complex carbohydrates. Uh, so they can fix the carbon dioxide from the atmosphere, and then those organic carbon sources can be used by the fungus being a heterotroph. Fungus can't make its own food, so fungus can use those organic carbon. And what is the algae getting? The fungus, the mycelia of the fungus, remember, that is providing a space for the algae to grow. Okay, so fungus is providing a substrate to the algae to grow, and algae is providing food to the fungus. Okay, so it's a symbiotic relationship. Mycorrhiza we have already discussed, rhizobium also we have already discussed. So in the roots of the leguminous plants, there are structures called root nodules, and inside those nodules, these bacteria live, and they enable the nitrogen fixation. Okay, any doubt? I hope this is clear. If anybody has any doubt, please let me know. Okay. Moving on to the next question. Which of the following is correct reaction representing the denitrification process? We have, again, I have already shown you and explained you all the process, so all of you should write the answers. Which of the following uh, is the correct reaction representing the denitrification process? Option A, ammonia to nitrite. Option B, nitrite to nitrate. Option C, nitrate to nitrogen. Option D, nitrogen to ammonia. Which is the denitrification process among these? See, I'm asking the same question in different styles so that you remember which reaction is what. This is very important topic. So all of you try to answer this. I've already explained all the process. So this is something I've just told you. What would be the correct answer? Again, only 3 people have answered. What about others? Anyone else wants to answer? Four of you are saying option C, nitrate to nitrogen. That is the correct answer. So the first and the second options are basically the nitrification process. That's option C is denitrification process, which involves the reduction of nitrate to nitrogen. Option D, nitrogen to ammonia. This is called nitrogen fixation. Okay, so now all, I hope this is clear to everyone. Whoever had still any doubt, I hope now it is clear. So we have nitrification first two, then denitrification, and then nitrogen fixation. If still anybody has doubt, please ask. I can explain as many times as you want, but you need to understand this. Moving on to the next question. Under anoxic condition, green and purple sulfur bacteria can utilize dash as a source of energy. Under anoxic conditions, green and purple sulfur bacteria can utilize dash as a source of energy. What should be the source of energy? Option A, nitrogen. Option B, oxygen. Option C, carbon dioxide. And option D, light. All of you write your answers in the chat box quickly. Under anoxic condition, green and purple sulfur bacteria can utilize dash as a source of energy. Nitrogen, oxygen, carbon dioxide, light. What would be the answer? Anyone else? I have one answer in the chat box. Anyone else? What is the source of energy that is used by green and purple sulfur bacteria? I think we have discussed this in the previous session when we were discussing Vinogradsky column. Okay. I see all of you are answering option D, light. That is the correct answer. So green and purple sulfur bacteria, they can utilize light to reduce carbon dioxide to carbohydrates using H2S instead of water. So basically, since it's an anoxic process, so remember electron transport chain. So in the electron transport chain, what was the terminal electron acceptor? The terminal electron acceptor was H2O or oxygen, right? So instead of H2O now, under anoxic condition, it will use H2S to accept the electrons. Okay, rest all the process will be same. The electron acceptor would be H2S. And what is this bacteria called? It is utilizing light as energy source and carbon dioxide, uh, using carbon dioxide to make carbohydrate. So what should it be called? What type of organism is this? What type of bacteria is this? Anybody?
Ma'am, could you please say it again?
These bacteria are using light as the energy source and carbon dioxide as the carbon source directly. So what should we call them? What kind of troughs?
Autotroph.
Yes, autotroph. And what would be the prefix?
Autotrophs.
Sorry?
Is it
Photo.
photo?
Photoautotrophs, yes. So because it is using light as the energy source, it is the prefix photo will be there. If it is using chemical energy, then it would be chemo. Okay. And autotroph, if it is using carbon dioxide as the carbon source. Heterotroph, if it is using organic carbon as the carbon source. Okay. So these are something that we have already uh, discussed in the previous session, but since now we are at the towards the end of the course, we should be able to revise the previous things that we have already learned and connect it with the new things that we are learning. Okay, so do not forget the topics that have been covered in the previous session as we are going along. Okay, so it's a photoautotroph, that is correct. And these green and purple sulfur bacteria, they can grow under anoxic conditions. So if you remember the Vinogradsky column that we discussed in the previous session, in the anaerobic water zone, the middle part, where some amount of light is penetrating but not much oxygen can diffuse, in that area these can grow and they can use the H2S that was being liberated from the sediment part, right? Okay. Moving on to the next question. During anaerobic decomposition, complex polymers are broken into simple monomers in which of the following step? During anaerobic decomposition, complex polymers are broken into simple monomers in which of the following step? Option A, methanogenesis, option B, acetogenesis, option C, fermentation, and option D, hydrolysis. During anaerobic decomposition, complex polymers are broken into simple monomers in which of the following step? Methanogenesis, acetogenesis, fermentation, hydrolysis. All of you write your answers in the chat box. I have only one answer. Anyone else? Okay. Three of you have answered option D, hydrolysis. Let's see the correct answer. That is the correct answer. So these complex polymers, they are broken down into monomers by the process of hydrolysis. Basically, what are these complex polymers? These are basically the macromolecules that we have inside our body: carbohydrates, proteins, fats. So carbohydrates, they are basically polymers of sugar. Okay, then proteins, they are polymers of amino acid. Then fats are polymers of fatty acids. So they are the first step of degradation of any of these complex biomacromolecule or complex polymer is the uh, hydrolysis of these polymer into monomers. So carbohydrate will be broken down into sugars, proteins will be broken down into amino acids, and fats will be broken down into individual fatty acids. Okay, So this process is called hydrolysis, and different hydrolytic bacteria and cellulolytic bacteria are present that can perform this function. There are certain enzymes that are present in these bacteria that can perform these functions. Okay, so to break down the bond between two amino acids that are making the protein, like what is the bond called? It's called a peptide bond. Okay, and to break that peptide bond, you need some form of enzymes, and those enzymes are generally present in different organisms that can perform this hydrolysis. Once hydrolysis is done, these monomers can then be fermented into different steps, like either it can be fermented into carbon dioxide completely, which can then be converted into acetate, or some can be directly converted into acetate, and some can be fermented into propionate and butyrate, and which can ultimately go to the acetate. Once all these processes lead to acetate formation, this acetate is used by the bacterial group called methanogens, which can perform a process called methanogenesis, where they can use these acetate to form methane and carbon dioxide. Okay, so that process is called methanogenesis, and the bacteria that perform that are called methanogens. Okay, any questions or any doubts? Moving on to the next question. Methanogens belong to which of the following domains? Option A fungus, option B eukarya, option C archaea, and option D bacteria. Methanogens belong to which of the following domains? Fungus, eukarya, archaea, bacteria. Write your answers in the chat box. So I'll give you a hint. I've already told you that it belongs to bacteria, but which bacteria is also further divided into two domains, right? So which domain of bacteria? None of you knows the answer? Okay, I can see three of you have answered option C, archaea. That is the correct answer. So methanogens can produce methane during metabolism. So I already shown you in this figure from the acetate by methanogenesis. Methanogens can convert acetate into methane and carbon dioxide, and these methanogens, they belong to archaea domain of bacteria. So bacteria are divided into two categories, eukarya and archaea. Eubacteria and archaebacteria, or sorry, not eukarya, eubacteria and archaebacteria. So generally eubacteria are commonly written as bacteria, and archaebacteria are considered as a different subdomain, because all the archaebacteria, they most of the archaebacteria, they have the ability to grow in extreme environmental conditions. Okay, so methanogens are come under the archaea domain of the bacteria. Okay, any doubt or any question in this? Okay, moving on to the next question. What is the recommended period of time for a BOD test? What is the recommended period of time for a BOD test? Option A 1 day, option B 5 days, option C 2 days, option D 7 days. What is the recommended period of time for a BOD test? Okay, I already have three answers here. Option B 5 days, that is the correct answer. So the recommended period for the standard BOD test is 5 days. Okay, so BOD is biochemical oxygen demand. So whenever we have to test a water sample and check the how much the BOD of any sample is, uh, we need to wait for 5 days for the uh, complete procedure. Okay, so this is just a theoretical question. Moving to the next question. Dash is the largest source of SO2 in the atmosphere. Dash is the largest source of sulfur dioxide in the atmosphere. Option A burning of fossil fuels. Option B locomotive ships and vehicles. Option C volcanoes. And option D extraction of metals from ores. What would be the correct answer? All of you write your answers in the chat box. I can see two answers, option C and option A. Option C volcanoes and option A burning of fossil fuels. The correct answer is burning of fossil fuels, option A. So the largest source of SO2 in the atmosphere is actually burning of fossil fuels. Burning of fossil fuels release sulfur dioxide in the atmosphere uh, in the largest quantity. Okay. Okay, the next question is SO2 released into the atmosphere is converted into dash and dash. So now you have seen the source of the SO2. Now next question is SO2 released in the atmosphere is converted into dash and dash. Option A sulfur and oxygen. Option B sulfuric acid and sulfates. Option C hydrogen sulfide and oxygen. Option D sulfonic acid and oxygen. Sulfur dioxide released into atmosphere is converted into dash and dash. Sulfur and oxygen, sulfuric acid and sulfates, hydrogen sulfide and oxygen, sulfonic acid and oxygen. What would be the answer? All of you write the answers in the chat box. SO2 released in the atmosphere is converted into dash and dash. It is converted into two compounds. What would be the answer? Okay, I have three answers in the chat box. Four. Anyone else wants to answer? Four of you have answered. Anyone else? Okay. I can see most of you are answering option B. sulfuric acid and sulfates. That is the correct answer. So sulfur dioxide, once released into the atmosphere, it is converted into sulfuric acid and sulfates, and these sulfuric acids and sulfates are the reason for the acid rain that we have. So you remember, in case of nitrogen cycle we had nitrates, right? And in case of sulfur cycle we have sulfates. So all these nitrates and sulfates can ultimately lead to nitric acid and sulfuric acid formation. So when it rains, it can all these acids, they can actually mix with the rainwater, and that's how we are getting acid rains due to the pollution. Okay, so sulfur dioxide and like sulfuric acid and nitric acid, these are the main acidic components in the acid rain. Okay, I'll discuss the whole cycle, sulfur cycle, in one of the upcoming slides, so there I'll explain. Yeah, Mobani, do you have a question?
Yes, ma'am.
Yeah.
Ma'am, in question number 9, sometimes we see, yes, ma'am.
Yeah.
Sometimes we see that BOD 7 also happens. So, ma'am, am I right?
Uh, yeah, but mostly according to the guidelines, national guidelines, we do it in 5 days.
Okay, okay, okay.
Okay.
Yes, ma'am.
Yeah. Anything else? This is clear? Anyone else has any question? Please ask. Okay, then move on. Let's move on to the next question. Question number 12. Biogas generated at the end of the anaerobic digestion is mainly composed of dash and dash. Biogas generated at the end of the anaerobic digestion is mainly composed of dash and dash. Option A, ammonia and methane. Option B, H2S and CO2. Option C, CH4 and CO2, that is methane and carbon dioxide. And then option D, methane and carbon monoxide. Sorry. Biogas generated at the end of the anaerobic digestion is mainly composed of dash and dash. Ammonia and methane, H2S and CO2, CH4 and CO2, and CH4 and CO. All of you write your answers in the chat box. I can see already 5 answers are there. Anyone else wants to write their answers? Okay, so 5 of you who have answered are saying option C, methane and carbon dioxide. That is the correct answer. So, if you remember the methanogens, the process of methanogenesis, that was generating this methane and carbon dioxide, right? So, that process, these two gases that are formed at the end of that methanogenesis process are called biogas. And methanogens generally reside in the gut of certain ruminant animals, and then like cows and buffaloes, all the cattles mainly. So their gut has these methanogen bacteria, and these methanogens perform the anaerobic digestion of food that can lead to biogas formation. Biogas is basically the methane and carbon dioxide that is produced. Okay, so anaerobic digestion of biogas involves microorganisms breaking down the organic matter. So mostly these cattle are eating grass and plant sources. So they are complex carbohydrate sources. They are broken down. They're basically, those are organic carbon sources. So they are broken down into methane and carbon dioxide gas. Okay. And that's why this biogas is used as a fuel source. So if you remember, like if you have seen uh, anywhere, cow dungs are used for generating fuel while cooking. Okay, so that cow dung, using cow dung as a fuel source is because they have this biogas, methane and carbon dioxide, which can be used as a fuel source. Okay, so this we have already explained. Any questions? Okay, let's move on to the next question then. Which enzyme can catabolize starch? Which enzyme can catabolize starch? Option A, cellulase. Option B, dextranase. Option C, chitinase. Option D, amylase. Which enzyme can catabolize starch? Cellulase, dextranase, chitinase, amylase. Catabolize or catabolism means breaking down. Okay, so which enzyme can break down starch? Cellulase, dextranase, chitinase, amylase. What would be the correct answer? Write your answers in the chat box. Okay, I can see option D in the chat box. All of you have answered option D, amylase. That is the correct answer. So amylase catalyzes the hydrolysis of starch. So again, starch is a polymer. So remember, the catabolism of any complex polymer was done by a hydrolysis reaction. So amylase is one such enzyme that can do the hydrolysis of starch. Starch is a carbohydrate basically. Okay, so starch is made of uh, glucose. Glucose is the monomer unit of starch, and they are joined by alpha 1,4 glycosidic bonds. So I have discussed about glycosidic bonds in one of the previous sessions. So I hope you know what is alpha 1,4 and beta 1,4 glycosidic linkages. So starch is made of glucose, which are connected by alpha 1,4 glycosidic linkages, and these bonds are broken down by uh, the enzyme amylase. Okay, they can hydrolyze these alpha 1,4 glycosidic bonds. Okay, any question or any doubt? So this is how a starch molecule looks. So this is the alpha 1,4 linkages, and these are alpha 1,6 linkages. That is a different linkage. So both of these linkages are broken down. So the breaking of these bonds are called hydrolysis of these bonds. Why hydrolysis? Because the H2O is involved in the breaking down. So once this bond is broken down, H and OH is added to either side of the bond. So here the H will go and here the OH will go. So that's how we have. So any hydrolysis reaction will always have a H2O molecule that will help in that breaking of bond. Okay, so if H2 is involved in the breaking of bond, it is called hydrolysis, and this is the breaking of starch into individual glucose units by the enzyme amylase. Any question? I hope that's clear. Moving on to the next question, question number 14. Fatty acids are metabolized through which of the following processes? Fatty acids are metabolized through which of the following process? Option A, alpha oxidation. Option B, beta oxidation. Option C, hydrolysis reaction. And option D, oxidative phosphorylation. Fatty acids are metabolized through which of the following process? Alpha oxidation, beta oxidation, hydrolysis reaction and oxidative phosphorylation. What would be the correct answer? All of you write your answers in the chat box. I can already see 4 answers in the chat box. Anyone else wants to write the answers? Okay, so 4 of you are saying option B, beta oxidation pathway. That is the correct answer. So fatty acids are metabolized by this beta oxidation pathway. What is this beta oxidation pathway? So fatty acids are basically long chain aliphatic or alkane chains are there, long alkane chains with at the end of which has a carboxylic acid group. So a long carbon chain with COH at the end. That COH will be replaced by coenzyme A. So we'll have CO and C double bond O, and then coenzyme A will be added. After the addition of coenzyme A, this fatty acid can undergo beta oxidation, where, so what do we mean by beta? Beta is the position of the carbon. So if this is the functional group, then the carbon at nearest to the functional group would be alpha. So this carbon would be alpha, this will be beta, this will be gamma, and so on. Okay, so the cutting will be near the beta carbon. So that's why it is called beta oxidation, because this carbon will get oxidized, beta carbon will get oxidized. Okay, so right now how many carbons do we have in this chain? 1 2 3 4 5 6 7 8 9 10 11 12 13 and 14. Okay, so 14th carbon is basically the one that is carrying the coenzyme A. In beta oxidation, the beta carbon is getting oxidized and 2 carbons will be released. So if this part is cut, these 2 will be released, and then from 14 now we'll have 12 carbon chain with a coenzyme A attached to the final carbon. So this was the beta carbon that we had here, which now has the C double bond O and coenzyme A, and this 2 carbon molecule that is released, that is the acetyl coenzyme A. Okay, so again this part will again undergo another cut, another beta oxidation cycle. One more molecule of acetyl coenzyme A will be released, and we'll have 10 carbon chain. Then again we'll have 8 carbon chain, and one more acetyl coenzyme A will be released. And as you go along this cycle, ultimately you will have all the whole chain will be converted into acetyl coenzyme A, and you know where acetyl coenzyme A goes in a biochemical pathway. Okay, where does it enter? Which cycle does acetyl coenzyme A enter? When we discuss the biochemical cycles, remember acetyl coenzyme A was the one that was entering one of the cycles. Which was that? One of the pathways or one of the cycles. You remember, anybody? Yeah, Rudraditya have answered Krebs cycle. That is the correct answer. So acetyl coenzyme, all the acetyl coenzyme from the fatty acid oxidation will enter the TCA cycle. Mostly they will enter the TCA cycle. Some of them can have other roles as well. Okay, so you can now relate. There we only studied about glycolysis and then TCA cycle. So glycolysis was from the carbohydrate source. Basically carbohydrates will be broken down into sugar, which will ultimately lead to glucose formation, and glucose will be converted into pyruvate, which can form acetyl coenzyme that can enter the TCA cycle. But apart from carbohydrate, we eat other things also, right? We eat protein, we eat fats. So all of these things will have a way to enter the Krebs cycle and electron transport chain at the end, because at the end whatever we are eating is going to get incorporated into the ATP generation process. Okay, so this is clear how fatty acid oxidation pathway, fatty acid metabolism is again connected to the previous pathways that we have studied. Okay, the next question is the following sentence true or false? Fatty acid metabolism is an ATP utilizing process. Fatty acid metabolism is an ATP utilizing process, true or false? Tell me, fatty acid metabolism is an ATP utilizing process, true or false? Sorry. It's a fact. It's a ATP utilizing process or ATP generating process. Think. Is it using ATP or is it making ATP? You just saw the beta oxidation part. All of you are saying true. It's an ATP utilizing process. It's actually false. It's a ATP generating process. So beta oxidation generates ATP. Okay. So a thumb rule is any catabolic process generates ATP. Okay. So fatty acid metabolism is basically fatty acid breakdown. Any breakdown process generally generates ATP. Any synthesis process will require ATP. So fatty acid synthesis will require ATP. For making fatty acids in our body will need energy. When the body is breaking down fats, it is generating energy. Okay, it is generating ATP molecules. So fatty acid oxidation, it's a highly ATP generating process. Okay, so fat breakdown actually generates a lot of energy, significant amount of energy. Is this clear? Since all of you said true, I
Ma'am,
hope.
but here
Yeah.
metabolism is mentioned, so how we will understand, is it forming or getting broken down?
Uh, okay, your point is right somehow, but since in the previous question we did discuss about beta oxidation, the questions were in that flow. Okay, your point is not completely wrong. So metabolism here, the question should have been fatty acid catabolism or fatty acid oxidation. Okay. But even if metabolism is written, then how did you say it's true? Because metabolism can also mean catabolism. So then in that case, it should have been like both ATP generating and ATP utilizing. The synthesis part will utilize energy, and the oxidation part or the catabolizing part will be uh, generate will generate ATP. Okay, generate energy. I hope that's clear.
Yes, ma'am.
Okay. Okay, let's move to the next question. The range of total Jaldal nitrogen in sewage is normally around. The range of total Jaldal nitrogen in sewage is normally around. Option A 10 to 15 mg per liter, option B 20 to 80 mg per liter, option C 30 to 50 mg per liter, and option D 5 to 20 mg per liter. the range of total gel-dal nitrogen in sewage is normally around? Write your answers in the chat box. I can see 4 of you have answered option B, 20 to 80 mg per liter. Let's see the answer. That is the correct answer. So total gel-dal nitrogen is basically the total organic nitrogen present in the sewage. So the range is between 20 to 80 mg per liter. So that much organic nitrogen. So what do we mean by organic nitrogen? So any ammonia or ammonium ion form. Okay. So inorganic nitrogen is the N2 nitrogen that is present in the atmosphere in the gaseous form. So N2 is the inorganic nitrogen which is fixed into organic form, that is ammonia or ammonium. Okay, so that is the organic nitrogen content in the sewage, which is named as total gel-dal nitrogen, TKN, and the range of TKN is generally around 20 to 80 mg per liter in sewage. And this factor is monitored when the sewage water is treated, when the wastewater treatment goes along. At that time, like how BOD is monitored, TKN is also monitored. Okay. Next question is, according to the new National Green Tribunal guideline, what is the maximum BOD3 value at 27 degrees Celsius for treated wastewater being discharged into any inland surface water body? According to the new National Green Tribunal guideline, what is the maximum BOD value for treated wastewater being discharged into any inland surface water body? Option A 80 mg per liter, option B 30 mg per liter, option C 10 mg per liter, and option D 5 mg per liter. What would be the correct answer? What should be the maximum BOD value that can be discharged in the inland surface water body? All of you write your answers in the chat box. I can see two answers, option C and option B. Anyone else wants to answer? Let's see the correct answer. The correct answer is option C, 10 mg per liter. So earlier it was 30 mg per liter, but now the new guidelines have reduced it to 10 mg per liter. Okay. So 10 mg per liter is the revised sewage discharge standards for the wastewater being discharged in the mega and metropolitan cities. In India, mega cities are defined as the cities with population greater than 10 million, and metropolitan cities are defined as the population greater than 1 million. Okay. So mega and the metropolitan city, this is the BOD that can be BOD limit that can be released or discharged into the water bodies. Okay. In the class 2, so there are different classes for the cities, right? So in the second category, you can see the range is slightly higher. Since more people are residing in these cities, mega and metropolitan cities, the limit has been reduced, and for the other areas, the limit has been decided based on the population. Okay. Next question is, ruminant digestive system is analogous to. I've already explained this. Ruminant digestive system is analogous to option A, aerobic digestion tank, option B, anaerobic digestion tank, option C, trickling bed reactor, and option D, continuous stir tank reactor. A ruminant digestive system is analogous to. Write your answers in the chat box. I've already explained this, so everybody should be able to answer. Remember when we were talking about biogas? Okay, I can see 4 of you have answered option B, anaerobic digestion tank. That is the correct answer. So we have already discussed about biogas formation. So the digestive system of these animals, they have these methanogenic bacteria. Like our gut has some bacteria. Similarly, these cattle also have certain type of bacteria that are residing inside their gut. Okay, and when they eat these uh, plants and grass, so all these are different carbon sources, right? So these carbon sources are then, or organic sources are then broken down into methane and carbon dioxide, that is uh, the biogas. And the environment is anaerobic, so it is similar to the anaerobic digestion tank. What is anaerobic digestion tank? It is actually the tank that is used in the sewage treatment plant. So when sewage is treated first, all the particulate matter and everything is filtered out, and then it is sent to an anaerobic digestion tank where the organic matter in the sewage can be broken down into methane and carbon dioxide into biogas by these uh, methanogenic bacteria, methanogens, and that is basically what is happening inside the gut of these cattle also. Okay, so anaerobic digestion tank is analogous to the ruminant digestive system. So same thing is written. Microbes digest and ferment the plant material to provide energy to the ruminant, and in the process, gases like carbon dioxide and methane are released. That's clear? Any doubt? Moving on to the next question. Which of the following sources is responsible for the most amount of methane emitted globally? Which of the following sources is responsible for the most amount of methane emitted globally? Option A, coal mining, option B, rice cultivation, option C, wastewater, and option D, enteric fermentation. Coal mining, rice cultivation, wastewater, and enteric fermentation. Which of the following is the source of methane, most amount of methane that is emitted? I already have 2 answers in the chat box. What about others? All of you write your answers in the chat box. Coal mining, rice cultivation, wastewater, and enteric fermentation. Anyone else wants to answer? Okay, 2 of you who have answered option D, enteric fermentation, that is the correct answer. So that is the source of methane. I told you in the previous slide only, the digestive system or the anaerobic digestion tank they generate the methane, right? So the digestive system of the ruminants, they generate the most amount of methane that is emitted globally. Okay. So enteric fermentation is a part of digestive process of in the ruminant animals, where microbes in the digestive tract decompose and ferment food, producing methane as a byproduct. Okay. Question number 20. Which pair of enzymes catalyze oxidation of hydrocarbons under aerobic conditions? Which pair of enzymes catalyze the oxidation of hydrocarbons under aerobic condition? Option A lipase, option A lipase and amylase, option B cellulase and hydrogenase, option C monooxygenase and dioxygenase, option D ligase and lipase. Which pair of enzymes catalyze the oxidation of hydrocarbons under aerobic conditions? Lipase and amylase, cellulase and hydrogenase, monooxygenase and dioxygenase, ligase and lipase. What would be the correct answer? All of you write your answers in the chat box. Okay, I see 4 of you have answered. Anyone else wants to answer? Okay, 4 of you are answering option C, monooxygenase and dioxygenase. That is the correct answer. So these are the two enzymes that help in the catalysis of hydrocarbon oxidation under aerobic condition. So this is how an aliphatic hydrocarbon is oxidized. So aliphatic hydrocarbon like octane or any alkane, they will first be converted into alcohol by monooxygenases, and then that alcohol will be further oxidized into aldehyde and then carboxylic acid. The carboxylic acid of an alkane, I have already told you what is it. It is a fatty acid, and then that fatty acid can undergo beta oxidation. Okay, so the enzyme that is involved in the oxidation of aliphatic hydrocarbons are the monooxygenases. There are some aromatic hydrocarbons also like benzene. So how the benzene will be oxidized? For oxidation of benzene monooxygenase will act on it and add one OH. Okay. Again, one more benzene monooxygenase will add one more OH, and then you will have two OH added to the benzene, which will be the catechol. The product is called catechol. This catechol can be oxidized by catechol dioxygenase. So this is class of uh, enzyme that is monooxygenase. So monooxygenase can add OH, one OH at a time, whereas dioxygenase can oxidize two OH at the same time. Okay, so catechol 1,2 dioxygenase can actually oxidize these two OH groups that are present in the catechol together to form carboxylic acid groups. Okay, so alcohol is always oxidized into aldehyde and carboxylic acid. Okay, so you can see here, so cis muconate is formed from catechol by catechol 1,2 dioxygenase. These are just examples. So some aromatic hydrocarbons. So another aromatic hydrocarbon is toluene. Toluene basically benzene with a methyl group substituted on one of the carbons. So methyl group on the benzene. Now when toluene is oxidized by a dioxygenase, now since it is a dioxygenase, in one step process we will have 2 OH added. In case of monooxygenase, it was a two-step process because two steps are required for monooxygenase to add, because in one step it can add only one OH. This toluene dioxygenase, it can add 2 OH at the same time in one step. These 2 OH will then get further oxidized into carboxylic acid groups. Okay, so once 2 OH groups are added, this is called catechol. So and then that catechol methyl catechol will be there. So this is catechol and one methyl group is there. So methyl catechol. So methyl catechol dioxygenase will further oxidize these OH into carboxylic acid groups. Okay. Is this clear? If you have any question or any doubt, please let me know. During the lightning, the high temperature and pressures created in the air converts nitrogen into dash. During lightning, the high temperatures and pressures created in the air converts nitrogen into dash. Option A chloroform, option B ammonia, option C oxides of nitrogen, and option D oxygen. Chloroform, ammonia, oxides of nitrogen, and oxygen. During lightning, high temperature and pressure created in the air converts nitrogen into dash. What would be the answer? All of you write the answers in the chat box. I can see 4 of you have answered option C. Anyone else wants to answer? Option C, oxides of nitrogen. That is the correct answer. So nitrogen is converted into oxides of nitrogen directly by lightning, because lightning has a high temperature and pressure that can directly convert the nitrogen into nitrates or oxides of oxides of nitrogen are basically NO2, NO3 minus, NO2 minus, and that those oxides of nitrogen can dissolve in water and come down as rain, which is called acid rain. So I told you how sulfates mixing with water were generating sulfuric acid. Similarly, oxides of nitrogen with water, they will form nitric acid, and that will cause acid rain. So this is the whole nitrogen cycle that I have been talking about in the previous slides as well. So you have atmospheric nitrogen, which is fixed by these bacteria, and once nitrogen fixation occurs, the product is ammonia. Okay, ammonium is then oxidized into nitrite and nitrate by the process of nitrification, and ammonia to nitrite is nitrifying bacteria. Ammonia to nitrite and nitrate, that these are the process is nitrification, and the bacteria that catalyzes the reaction are called nitrifying bacteria. These nitrates are then again by the process of denitrification, they go back to nitrogen, atmospheric nitrogen, uh, and these atmospheric nitrogen again gets converted into ammonia by nitrogen fixation. So this is the whole cycle that goes on. Okay, any question or any doubt? I've already explained this multiple times, so I think this is clear. If still anybody has any questions or any doubt, please ask. Okay, moving on to the next question. Which of the following is a sedimentary type of biogeochemical cycle? Which of the following is a sedimentary type of biogeochemical cycle? Phosphorus and carbon monoxide, oxygen and nitrogen, phosphate and nitrogen, phosphate and sulfur. Which of the following is a sedimentary type of biogeochemical cycle? Phosphorus and carbon monoxide, oxygen and nitrogen, phosphate and nitrogen, phosphate and sulfur. Write your answers in the chat box. I can see 4 of you have answered option D, phosphate and sulfur. Anyone else wants to answer? Okay, let's see the correct answer. The correct answer is phosphate and sulfur, option D. So what do we mean by sedimentary type of biogeochemical cycle? The cycles where the elements are originating and coming back to the crust of the earth. Okay, so Like in nitrogen cycle, the nitrogen is going into the atmosphere. Same for oxygen, oxygen is in the atmosphere. Carbon dioxide, that is also in the atmosphere. Carbon monoxide, carbon dioxide, that also goes in the atmosphere. But phosphorus and sulfur, they are present in the sediment of the earth or crust of the earth. Okay, so the cycle where the elements are recycled into the crust of the earth, that those are called sedimentary type of the biogeochemical cycles. Okay, next question. Aerobic oxidation of H2S is carried out by which of the following bacteria? Aerobic oxidation of H2S is carried out by which of the following bacteria? What would be the answer? Aerobic oxidation of H2S is carried out by which of the following bacteria? Option A, purple sulfur bacteria. Option B, green sulfur bacteria. Option C, Thiobacillus and Beggiatoa. Option D, D-sulfur vibrio and D-sulfobacter. All of you write your answers in the chat box. I can see 3 of you have answered already. Anyone else wants to answer? Write the answers in the chat box. Okay, all 3 of you are saying option C, Thiobacillus and Beggiatoa. That is the correct answer. So Thiobacillus and Beggiatoa, both of them can oxidize H2S aerobically. So this is the whole sulfur cycle. So we have been talking about nitrogen cycle uh, till now. So this is similar to that in some ways. So like elemental nitrogen, elemental sulfur is again oxidized into SO4 2 minus, that is sulfates. These sulfates are then either assimilated into the proteins, the SH group of proteins. You know which amino acids have SH or sulfur in their side chain. Cysteine and methionine have sulfur in their side chain. Okay, so proteins containing these amino acids will need sulfur. So this sulfate is assimilated in those proteins. The sulfur from these sulfates is assimilated into those proteins, and later when proteins are broken down, the sulfur is again desulfurylated and goes to the H2S. So this is one process. These sulfates are directly reduced into H2S by certain bacteria, which are sulfur reducing bacteria, D-sulfovibrio and D-sulfobacter. They can reduce these sulfates into H2S directly. Okay. And these H2S can then get oxidized into elemental sulfur by bacteria like Thiobacillus and Beggiatoa, and this process is aerobic. Okay, now similar process is undergoing in an anaerobic manner also. So there are elemental sulfur that are present, which are anaerobically converted into sulfates by purple and green phototrophic bacteria. Remember, we have talked about this purple and green sulfur bacteria that can use the elemental sulfur and oxidize into sulfate. Okay, so these are anoxygenic bacteria. They can do the oxidation in the absence of oxygen, and they can also oxidize H2S into elemental sulfur. So this is two-step of oxidation. If you see here, from H2S, the sulfur is oxidized into elemental sulfur, and then it is oxidized into sulfates. Okay, why everything is coming ultimately to the most oxidized form of sulfur, that is sulfate, because that can be used for the assimilation of sulfur into the proteins. Why do we need sulfur in our body? Because some proteins require sulfur in our body. So where we are going to provide the sulfur from? So this is how the sulfur cycle in the environment goes on, so that these sulfur can be assimilated in the proteins inside these bacteria. So in these bacteria are also using certain proteins, they are also having proteins in their system, right? So this is how the whole process goes along. So the elemental sulfur, either anaerobically or aerobically, they can be oxidized into sulfates. Anaerobically, the oxidation is carried out by purple and green sulfur bacteria. These are anoxygenic phototrophic bacteria. These are autotrophic, and they use light as their energy source. Okay, so they can oxidize H2S into elemental sulfur, and uh, then oxidize sulfur into sulfate, anoxygenic, anaerobically. And then aerobically, the H2S is oxidized into sulfur by thiobacillus and Beggiatoa, and the sulfate is reduced by desulfovibrio and H2S. There are desulfuromonas which can anaerobically reduce the sulfur into H2S. Okay. So when we create the Vinogradsky column, you can actually see the division. So thiobacillus and Beggiatoa will be there at the top part because they are aerobic. Then purple and green sulfur bacteria are present in the middle part where light can penetrate but oxygen cannot. And these desulfovibrio and desulfuromonas, they are present at the bottommost layer where there's no oxygen and light, and they can actually release H2S. That H2S will reach the middle portion, and these purple and green sulfur bacteria can use these H2S to make sulfates. Okay, so this is the last question of the session. Anybody has any questions or any doubts regarding?
Ma'am,
And yeah,
ma'am, could you please explain the sulfur cycle again in short?
yeah. So basically, two things are happening mainly. Okay, oxidation and reduction. So elemental sulfur, it is oxidized into sulfate, or H2S is oxidized into elemental sulfur, and then it is oxidized into sulfate. Okay, see it in this way. So H2S can be either oxidized aerobically by these bacteria or anaerobically by purple and green sulfur bacteria. Okay, then this elemental sulfur can further be oxidized into sulfate. So for aerobic oxidation, it can happen without the help of any bacteria. Also aerobic oxidation, air oxidation can happen for the sulfates. And for anaerobic oxidation, again purple and green sulfur bacteria can carry out this oxidation. So sulfur will be converted into sulfates. These sulfates will be assimilated into the proteins wherever the wherever sulfate assimilation is required, that will be happening from these sulfates. Sulfates can be reduced back to H2S by bacteria like desulfovibrio and desulfobacter, and this elemental sulfur can actually be reduced by desulfuromonas anaerobically, complete anaerobic process, and all of these will form this H2S again. So H2S will form sulfate, and sulfate will again come back to H2S. Okay, and the intermediate is elemental sulfur. Is this clear?
Okay, ma'am. Thank you, ma'am.
Yeah. Any other question? Any other doubt?
Ma'am, I have a doubt from week 11's question number 8
In
from the
your
assignment.
assignment. Okay, tell me.
Ma'am, the question is given alveolar sacs are colonized by dash and alveolar ducts are colonized by dash. The options are option A, coaxilla, chlamydia, option B, chlamydia, coaxilla, option C, Haemophilus influenza, streptococcus pyogenes, option D, staphylococcus aureus, mycobacterium tuberculosis.
Which question is this? Question number?
Question number 8 from week 11.
Okay. Alveolar sacs and alveolar ducts. Okay, so what is your doubt?
Ma'am, which one will be the correct option here? I have marked A, but I'm not sure of the answer.
Okay, your answer keys are not yet released, so I'm not really allowed to tell you the answers now. Can we discuss about this in the next class once your answer keys are out?
Yes, yes.
Because this is a theoretical question. If it was a numerical question, I would have helped you out. But okay, just wait for if you can check if your lecture videos have this, you can check it out. But wait for the answer key to released, and then I'll tell you. We'll discuss the questions in the next session. Next session will be a revision session, so whatever doubts will be there, I'll definitely help you out. Okay. Uh, okay. I need to ask you one thing. Tell me in which assignments the answer key is given wrong. So I remember from the radioactivity, one question was there. Can somebody write the question number and the assignment number in the chat box? If someone can do it quickly. I'll try to get in touch with the NPTEL team. If let's see if they correct it. Till then, anyone else has any other doubt in this today's lecture? So week 1, question 6. Week 1 also you have a doubt. Okay. Okay, I'll look into all these questions, all these assignment questions, and I'll get back to you in the next week. Okay, I'll try to get in touch with the NPTEL team if they change the answer keys, or then it would be good.
Okay, ma'am.
Okay.
Thank you, ma'am.
Yeah. Anyone else has any other doubt? Anything you want to ask? The next session will be a revision session for all the 12 topics that we have discussed. So it would be kind of a practice session for you for your exams, before your exams. Anything you want to ask, or any doubt, any questions? Okay, if there are no more questions, then I think I can end the session today. Thank you all for joining. I'll close the class now here. Okay, thank
Thank you,
you.
ma'am.
Yeah, bye bye.